Subtractive Proteomics for Identification of Drug Targets in Bacterial Pathogens, a review. Abstract: the first step in drug and vaccine discovery is the identification of the target. Subtractive proteomics can be widely used for the process. This approach has been used to find drug targets for the pathogen, which are resistant to drugs and for which there is no vaccine. This insilico method can reduce the cost and time for finding the drug targets. It involves subtraction of proteins of host and pathogen which provide a set of proteins, which are essential for pathogen but absent in gut flora and host. A review was carried out on a subtractive proteomics approach on 29 pathogens, as reported from 2008 to 2018. These organisms have been categorized under two categories: multiple drug-resistant organisms and those which have no effective drug to date. By subtractive proteomics, the total proteome undergoes a subtraction process at each step, to narrow down to few drug targets. Tools and databases for subtractive proteomics have also been described. Keywords: subtractive proteomics, drug targets, in silico, vaccine. Introduction. The genome sequencing projects of microbial pathogens have been growing rapidly in the past decade. We also have complete information on the genome of human beings. This has led to the development of new methods for studying the interaction between humans and microbial pathogens. This, in turn, has improved various tools to combat a broad range of pathogens. 1. The process of introducing a drug in the market still takes 10 to 15 years, despite the use of high-throughput techniques and synthetic chemistry. Hence, it increases the cost of developing a drug. 2. Using integrated genomics, transcriptomics, and metabolomics for target discovery in several diseases is a trend these days because it makes the entire process quick and cost-effective. Various bioinformatics tools have been developed, several of which involve a comparative analysis of host and pathogen genome, which reveal drug targets which are non-homologous to human and essential for pathogen survival. In silico subtractive proteomics is one such tool that is used to find novel drug target and vaccine candidates in pathogens, by comparing human and pathogen proteome and finding a protein which is essential for the survival of pathogen but absent in humans. 3. The current trends in target discovery for most human diseases are the use of computational approaches, with proteomics, integrated genomics, transcriptomics, metabolomics, interactomics, and signalomics, especially for infectious diseases, cancers, neuroendocrinal diseases, and cardiovascular diseases, thereby making the entire process of drug discovery faster and more cost-effective. Currently, to identify novel drug and vaccine targets, genomics, and specially in silico comparative, subtractive and functional genomics are widely used, especially to develop effective antibacterial agents and vaccines against bacterial pathogens that are resistant to existing antibacterial regimes, for which a suitable vaccine is not available. 4,5. Many diseases are caused by a bacterial pathogen. So many people die every year due to these diseases. Despite the presence of antibiotics, many resistant strains have developed which makes them difficult to control. The identification of drug targets is the first step in the drug discovery process. Due to the presence of the host and pathogen sequences, strategies have shifted from generic to genomic, proteomic, and metabolomics approaches to identify the novel drug targets. New drug targets are required for designing of new antibiotics against drug-resistant pathogens. Drugs being used currently to treat diseases caused by pathogens show less to more side effects in humans. Also, repeated use has led to the development of drug resistance in pathogens, so there is a need to find new drugs and drug targets, to be able to combat pathogens efficiently. A novel approach called subtractive proteomics has been found to be able to find novel drug targets in pathogens. 99. This is a step ahead towards developing new and effective drug targets. This review elucidates an overview of in silico subtractive proteomics approaches used to identify drug targets in various pathogenic bacteria. We have also discussed the advantages, disadvantages, and future prospects of this approach. 2 characteristics of a drug target. A drug target should be non-homologous to host and an essential protein for the survival of the pathogen. Although ideally, the target should have 4 properties: 1. It should be essential protein for survival and pathogenesis of target organism. 2. Target should be druggable. 3. Structural and functional characterization, with established assays for screening small molecule inhibition. 4. It should be different from current drug targets to avoid cross-resistance between them. 6. 3 strategies used. It has been shown in Figure 1. A. Identification of paralogous protein. Paralogous proteins are homologous proteins that have diverged within one species. CDHit, cluster database at high identity with tolerance, is a vastly used program for identification of paralogous proteins, which are finally excluded from the analysis 7. It is a widely used program for clustering genes/proteins to reduce redundancy during the process of identification of drug targets 8, 9, 10. The proteome of the pathogen is usually subjected to CDHit analysis with sequence identity cut off between 10 to 90% depending upon requirement 7. It is vastly accepted to keep sequence identity cut off 60% in order to maintain rigid criteria for removal of duplicate proteins 9, 11, 12. Proteins which are more than 60% identical are considered duplicate or paralog protein, which are excluded. The remaining non-paralog protein which has more than 100 amino acids taken for further analysis. It is usually assumed that protein which is less than 100 amino acids are less likely to be essential, hence excluded 13, 14. B. Essentiality analysis. Essential genes are those which are required for growth, adaptability, and survival of an organism. A deficiency of its protein can be deadly to an organism. Essential genes are most likely to be evolutionarily conserved across taxa and have a common function 15, 16, 17. The Database of Essential Gene, DEG, 18 is an online resource that contains information that essential genes in bacteria, fungi, plant, and animal, which are experimentally validated 19. The database can be analyzed for essentiality using DEG cutoff for e-value 10 to the power of 10th and a bit score greater than 100 is mostly used 20, 21 BLASTP against DEG is usually done to find essential proteins. The non-homologous proteins can be screened out. See identification of human non-homologues. BLASTP of Homo sapiens proteome with non-paralog protein is usually done with threshold expectation value 10-3 and bit score 100 to identify human non-homologous protein 14, 22, 23. Human homologs are excluded and the rest of the non-homologs are combined together for further analysis. This step is usually included to avoid cross-reactivity of drugs with a protein of host, to prevent its binding with the active site of host homologous proteins 24. Proteins having equals 35% and for which no hit was obtained, were called as host non-homologs 24. De-identification of orthologs in gut flora. Non-homology analysis with gut flora is essential. The gastrointestinal tract of healthy human houses about 1014 microorganisms 25. Gut microbiota lives in a symbiotic relationship with the host. They play an essential role in the metabolism of humans. They help in digestion of food particles and prevent pathogenic bacteria from growing inside gut 26. If we block the gut microbiota proteins, it may lead to an adverse effect on the host. In order to avoid such circumstances, proteins were subjected to homology search by performing BLASTP27 with gut flora proteome, keeping e-value threshold 0.000128. E-pathway analysis. Functional annotation of non-homologous essential proteins was done with their respective metabolic pathway in COS, KEG Automatic Annotation Server, 29 for specificity. The host and pathogen metabolic pathways can be comparatively analyzed using KEG, Kyoto Encyclopedia of Genes and Genomes, to find proteins involved in the pathogen-specific pathway for drug target identification. COS can provide functional annotation of genes through BLAST comparison with the KEG genes database. The result is KO assignments to metabolic proteins. It can automatically generate a pathway to those metabolic proteins. F. Drugability analysis. Drugability analysis of all shortlisted protein can be done by searching them in the drug bank database 30. All non-similar, essential, and hypothetical sequences of proteins can be screened by performing BLASTP against drug bank database 30. Drug bank database contains a number of protein targets with IDs of drug which are sanctioned by the FDA. Only those drug targets which have bit score greater than 100 and e-value 0.005 can be called drug targets. G. Virulence analysis. Bacteria, using virulence factors, degrade host defense mechanism with help of adhesion, colonization, and invasion, and thus cause disease. Vfdb is a database containing 4 virulence factors. These are offensive, defensive, nonspecific, and virulence-associated regulated proteins 31. The proteome of bacteria can be subjected to BLASTP against the Vfdb database. Cut-off bit score 100 and e-value 0.0001 is mostly used. H. Subcellular localization prediction. In microbes, proteins can be found at 5 subcellular locations. These are extracellular, periplasm, outer membrane, plasma membrane, and cytoplasm. Finding the subcellular location is essential for their categorization as a vaccine or drug target. Proteins in the cytoplasm can be called a drug target and those on the membrane can vaccine targets 32. A subcellular localization position can be obtained from the Uniprot database 33. But in the absence of experimental information, subcellular localization prediction can be done using Cello 34 and PSORTB 35. 4. Tools and databases used for genome subtraction. Swissport, TREMBL, and PIR protein databases have united to form Uniprot, Universal Protein Knowledge Base, Consortium. Uniprot is a comprehensive and rich database. It is also fully classified. It uses very accurately annotated protein sequence knowledge base, with extensive cross-references and query interface. TREMBL and Swissport are its 2 sections. TREMBL utilizes extensive cross-references and uses manually curated data at the same time Swissport utilizes manually curated data. It also provides many non-redundant sequence databases. The Uniprot databases consist of 3 database layers. I. The Uniprot archive, Unipark, which stores a complete body of publicly available protein sequence data and provides a non-redundant, comprehensive, and stable sequence collection. 2. The Uniprot knowledge base, Uniprot, provides a central database of protein sequence with rich, consistent, and accurate sequence and functional annotation. 3. The Uniprot NREF database, Uniref, provides data collection which is non-redundant and based on the Uniprot knowledge base in order to obtain complete coverage of sequence space at several resolutions. The proteome is a set of proteins expressed in a cell at a particular time by an organism. Most of Uniprot proteomes are based on the translation of a completely sequenced genome, and will normally include sequences that are derived from extrachromosomal elements like plasmid or organelle genome in an organism where these occur. Some proteomes may also include protein sequences based on high-quality cDNAs that cannot be mapped to current genome assembly due to sequencing errors or gaps. Uniprot proteome contains both manually reviewed, Uniprot KB/Swissport, and unreviewed, Uniprot KB/TREMBL, entries. The proportion of reviewed entries varies between proteomes and is obviously greater for the proteomes of intensely curated model organisms. There are about 12,274 reference proteome and 200,686 other proteomes. Several changes are often incorporated in Uniprot from time to time. The accession number has been expanded to help deal with the increase in sequences. Several protein identifiers and reference proteome have been included to deal with the overwhelming amount of sequencing data that is generated. Annotation scores have been included to identify the proteins with the highest level of functional characterization, which will greatly assist in comparative protein sequence analysis. We can search for proteins from Uniprot proteome 33. The genes that are mandatory to support cellular life are essential genes. These minimal gene set are needed for a living cell. Therefore, the functions encoded by these gene set are essential and can be said to be the foundation of life itself. 42,43. The definition of minimal gene set needed to sustain a living cell is of considerable interest as it has significance in practical use and it represents a fundamental question in biology. For example, most of the antibiotics target essential cellular processes, hence essential gene products of microbial cells are a promising target for antibacterial drugs. 44. DEG contains all genes that are essential and are continually updated. The proteins encoded by these essential genes are required for basic cell survival and thus present in all cells. A blast of the query sequence against DEG can yield homologous genes, which in turn can mean that query sequence is also essential. The essential gene can also be searched using function or name. All records in it can be browsed and extracted. Essential proteins are considered an excellent target for antibacterial drugs. DEG can be accessed online from its website for free. Its website is www.essentialgene.org. 18. DEG boasts of hosting currently available essential genomic elements records, in bacteria like archaea and eukaryotes containing protein-coding genes and non-coding RNAs respectively. Essential genes create a minimal genome, in a bacterium. They form a functional module set that plays a key role in synthetic biology, which is an emerging field. High-throughput sequencing and high-density transposon-mediated mutagenesis have led to significant advancement in research on essential genes under the diverse condition and revised essential gene concept, which includes non-coding RNA. Also 45. DEG10, which is a new release of a database of essential genes available at www.essentialgene.org, has been developed to accommodate these quantitative and qualitative advancements. In comparison to DEG 546, the number of bacteria with saturated genome-wide gene essentiality AHS nearly tripled in DEG 10, which has data for 31 bacteria. DEG 10 has nearly 12,000 bacterial essential genes, more than twice the number of those in DEG 5. Performing homologous searches with BLAST program 47 against DEG is common. 48 to 51, and so customizable BLAST tools have been developed. For the BLAST tools, one can perform BLAST search for a single gene, multiple genes, annotated genomes and unannotated genomes with filters to search to a subset of species or experiments with desirable p-values. A blast of DEG with the query sequence can give homologous genes, which will mean that query sequence is also essential. These essential proteins are excellent targets for antibacterial drugs. Essential genes can also be searched using function or name. All records can be browsed and extracted. Kyoto Encyclopedia of Genes and Genomes, KEG, can perform systemic analysis of gene functions in terms of a network of genes and molecules, and in that sense, KEG has a huge knowledge base. Pathway database is a major component of KEG. Pathway database comprises of graphical diagrams of biochemical pathways. It also includes almost all known metabolic pathways and few of the very known regulatory pathways. KEG has multiple objectives. First being the computerization of recent knowledge. This is the knowledge of biochemistry, genetics, and other disciplines. This would be done in terms of pathways of interacting molecules and genes. Second being, that KEG maintains a ligand database 52, 53, i.e. a catalog of chemical, compounds, and other substances in the living cell, which is linked to the towards predicting biological systems and provide with a new informatics technology. The fourth and final objective is that it can maintain a gene catalog. This catalog will be for all organisms with completely sequenced genome and will also include those selected organisms with partial genomes. KEG can be a very useful database. It can help to understand important biological systems, their services and functions, like in the cell, organism, and ecosystem in general. It can provide us with very molecular level information of datasets that are of large scale. These large-scale datasets are generated by genome sequencing and other high-throughput experimental technologies, and therefore this molecular level information is important. It is already established in the former paragraphs that KEGG has a huge knowledge base, and in that role, it can perform systemic analysis of functions that are done by genes, thereby linking this information obtained from genes with the functional information. Within the insilico subtractive proteomics, within the metabolic pathways in which they are involved, both host pathogen and pathogen common, the mapping of identified essential and non-human homologue proteins is done and is present in the KEGG database. It utilizes a comparative method of pathway analysis for both the pathogens and humans. The KEGG database is not only available online for free but is also regularly updated. www.genome.ad.jp web link, 36. Vfdb is a virulence factor database. It is comprehensive and user-friendly. It contains the latest existing knowledge about virulence factors from various bacterial pathogens. Bacterial pathogens pose a great threat to public threat, however greater studies on pathogenesis have enhanced our knowledge about disease mechanisms at the molecular level. Forming a database of virulence factors of major bacterial pathogens was necessary for future research. One can access the database using several keywords, etc. Blast search can be used against all VF-related genes. Vfdb works as a gateway for storing, searching, retrieving, and updating information about VFs from bacterial pathogens. We can access the database at www.mgc.ac.cn web link 31. Vfdb was constructed for twin purposes. To give us thorough and in-depth information about the main virulence factor of the best characterized bacterial pathogen. And second, was to explain the process of bacterial pathogenesis, thereby bringing in a new approach towards the treatment and prevention of infectious disease. In order to decode the potential novel or variant pathogens both in emergent outbreaks and in routine clinical practice, the entire genome sequencing is used. It is a big challenge though for the microbiologists or physicians to do an efficient characterization of pathogenomic composition, mainly because of their limited bioinformatics skills. Therefore it was introduced to Vfdb, an integrated and automatic pipeline, VF Analyzer, which systematically identifies known/potential VFs incomplete/draft bacterial genomes. Vfanalyzer first constructs within the query genome, orthologous groups, and analyzed reference genome from Vfdb in order to avoid potential false positives due to paralogs. Then it conducts sequence similarity searches from within the hierarchical pre-built datasets of Vfdb to precisely find untypical/strain-specific VFs. Eventually, the VF Analyzer achieves a relatively high specificity and sensitivity without any manual curation with the help of a context-based data refining process for VFs encoded by gene clusters. Apart from this, in order to make it easy for online analysis, an optimized interactive web interface is introduced to present its reports in a comparative pathogenomic style 54-57. Since the inception of Vfdb in 2004, it has always provided up-to-date knowledge of VFs from multiple medically important and significant bacterial pathogens. As such, it has become an all-in-one comprehensive online resource for finding information about the virulence factors of bacterial pathogens. There were twin reasons for constructing Vfdb. First, providing in-depth coverage, major virulence factors of bacterial pathogens with their structure features, their functions, and mechanisms used by these pathogens to allow them to win over new niches and to evade host defense mechanisms and eventually cause illness. Second, to provide knowledge of a wide variety of mechanisms, this helps in the prevention and treatment of infectious diseases. These are the mechanism used by researchers for bacterial pathogens to explain pathogenic mechanisms in bacterial diseases that are not yet characterized properly in order to develop new approaches. A bacterial pathogen is a bacterium that can cause disease, and its ability to cause disease is called pathogenicity. Virulence gives us a likelihood of causing disease, or a quantitative measure of the pathogenicity. Virulence factors are gene products that enable any microorganism to establish itself within a host and enhance its potential to cause disease. Virulence factor comprises of many things like bacterial toxins, cell surface proteins that mediate the bacterial attachment, cell surface carbohydrates and proteins that provide protection to a bacterium, and hydrolytic enzymes that add to the pathogenicity of the bacterium. The release, R1, is the core dataset of Vfdb. It included only the experimentally validated VFs of main medically important bacterial pathogens from 24 genera. Comparative genomic approaches were introduced in the release, R2, of Vfdb. It focuses on intra-genera comparison of complete genomes in terms of virulence apparatus. Apart from this, it also covers some genome-predicted VFS for comparative purposes. Release, R3, is dedicated to genetic diversity and molecular evolution of bacterial virulence factors. R3 is better than R2 as it performs intergenera comparative analysis. R3 includes many add-on VFs from the plant and animal pathogens, and sometimes, even from non-pathogens in comparison with previous releases. However, both are better extensions of the first release and are both based on the core dataset. Where R3 is VF-centered while R2 is genome-centered, I.E. both are extending in different directions. The release, R4, has helped in the improvement of twin aspects of infrastructural datasets of Vfdb. These are, I, removal of redundancy brought by previous releases, and also generated twin hierarchical datasets, one for experimentally verified VFs only and another consisting of full dataset including all the known and predicted VFs, 2, with the help of controlled vocabulary. Refining of gene annotation of the core dataset. The release, R5, to systematically identify known/potential VFs incomplete/draft bacterial genomes. The R5 includes an integrated and automatic pipeline, VFAnalyzer 54-57. It contains richly annotated drug targets and drugs. It contains wide-ranging data related to ontology, nomenclature, chemistry, structure, function, action, pharmacokinetics, pharmacology, metabolism, and pharmaceutical properties of molecule drugs both small and large. It also provides detailed information on the organism on which these drugs act, target diseases, proteins, and also genes. After 2 years of extensive manual annotation works, the database was made useful for wide-ranging omics, i.e. pharmacogenomics, pharmacoproteomics, pharmacometabolomics and even pharmacoeconomics. Drug Bank 3.0 was born. A blast search against the Drug Bank database reveals the druggable targets. It can be accessed from www.drugbank.ca30. It provides comprehensive data related to drugs with detailed drug target information. The Drug Bank database is a fairly detailed, and readily available, online database that contains information on various drugs and drug targets. Drug Bank can combine detailed drug target, i.e. chemical, pharmacological and pharmaceutical, data with comprehensive drug target, i.e. sequence, structure, and pathway, information, as it is both bioinformatics and a cheminformatics resource. Drug Bank is more analogous to an encyclopedia than a drug database, because of its broad scope, comprehensive and detailed referencing and a detailed data description that is unusual in nature. This is why links to Drug Bank are maintained for almost all drugs that are listed in Wikipedia. Drug Bank has wide uses for different fields, like in the drug industry, used by the medicinal chemists, by pharmacists, by physicians, by students, and the general public. Drug Banks has an extensive drug and drug target data that helps in discovering and repurposing of a number of existing drugs. Not only that, it also helps in treating rare and newly identified illnesses. The new version of Drug Bank, version 5.1.4, has been recently released on February 7, 2019 with 13,339 drug entries including a 2,595 approved small molecule drugs, B1,289 approved biotech, protein/peptide, drugs, C130 nutraceuticals and D over 6,304 experimental drug. This new version also contains additional 5,186 non-redundant protein, i.e. drug target/enzyme/transporter/carrier, sequences that are linked to these drug entries. Each and every drug card entry contains more than 200 data fields. Half of each of these data fields contains information being devoted to chemical/drug data, and the other half has information devoted to drug target or protein data. 58 to 62. If a blast search is done against the drug bank database, it reveals all the druggable targets. It can be readily accessed from the following link, www.drugbank.ca. 27. It has detailed and comprehensive data relating to drugs with detailed drug target information. 60. The biological sequence data size is rapidly growing due to genome projects and the emerging field of metagenomics. 63. High-throughput sequencing technologies are generating a huge amount of sequencing data, which has generated a need for bioinformatics tools for organizing and analyzing data. Mostly, biological sequences are related and can have homology, therefore clustering them in a group and finding consensus sequences can solve many sequence analysis problems. CDHit is a widely used program to cluster and compare large biological sequence data sets. CDHit stands for Cluster Database at High Identity with Tolerance. PHIT program is to remove paralogous sequences. It clusters and compares large biological sequence data sets. It clusters the protein clusters which meet user-defined similarity threshold, usually sequence identity. Each cluster has one representative sequence. The input is single file in FASTA format, whereas output is two files, a FASTA file of representative sequence and text files of the list of clusters. A CDHit suite has been launched, for clustering the user-uploaded sequence dataset or comparing to another dataset at different identity levels. A user-friendly web interface is provided by PHIT suite for clustering and comparing with added visualization tools. It also stores precalculated clusters for many public sequence databases. It is also routinely updated. 7. It accepts input in FASTA format sequence and output as non-redundant sequences. It only removes redundant sequences, and it does so by removing the overall size of the database without removing any sequence information. It can, from a given FASTA sequence database, produces a set of closely related protein families. The subcellular localization of a protein is closely related to its biological function. 64. Valuable information regarding its function can be obtained from the subcellular localization prediction of a protein. A gram-negative bacteria contains 5 main subcellular localization sites, i.e. the cytoplasm, the periplasm, the inner membrane, the outer membrane, and extracellular space. It has become extremely important to have an automated and accurate tool for subcellular localization prediction due to the increased number of genomic data. Based on N-peptide compositions, Cello uses the support vector machines trained by multiple feature vectors. There are 1,443 proteins within a standard dataset. It enables an overall prediction of 89%, a never-reported prediction rate. 37. Cello is an SVM classification system of the multi-class grade. It utilizes 4 types of sequence coding schemes: the amino acid composition, the dipeptide composition, the partitioned amino acid composition, and based on physicochemical properties of amino acids, sequence composition. 65. Computational prediction of subcellular localization is important as it provides information regarding its function. SORT BV 1.1 has a bacterial localization tool of the greatest precision. It cannot predict for the gram-positive bacteria but only the gram-negative ones. Thereafter modification was made in its v.2.0. This modified version has a precision of 96% for both grams-positive and negative bacteria. PSORT v.3.0 can return the associated probability value for each and a list of 5 localization sites. Above the cutoff value of 7.5, which is considered good, single localization can be assigned. Prediction of protein to be on the cell surface is of particular interest because such proteins can be primary drug and vaccine targets. A protein's subcellular localization gets influenced by several features that are present within the protein's primary structure, like membrane-spanning alpha helices, or the presence of signal peptide 38,66. PSORTB 3.0 continues to be the most accurate subcellular localization predictor, and it has increased recall and predictive coverage. It is the most flexible prediction software for prokaryotes, with both online web server and standalone software. This allows them to be incorporated into any bioinformatics pipeline. It has added the predictive capability of SCL prediction of archaeal protein, prediction for bacteria with typical cell morphologies, and addition of new predictive subcategories, which represents first SCL predictor designed to handle a diverse range of all prokaryotes and handle prokaryotic localization. 67. iBLAST. BLAST can search for regions of similarity between biological sequences. It calculates statistical significance and can compare nucleotide or protein sequences to a sequence database. Its algorithm is quite simple and robust, so much so that it can be implemented in a number of ways and in varied contexts that extend to include protein sequence database searches, direct DNA, motif and gene identification searches. With the help of heuristics, it can produce faster results. It also has the capability to estimate how many matches have occurred a given score by chance, by calculating an expect value, which eventually helps in judging the amount of confidence to have in the alignment. BLAST tool has been used to find essential sequences non-homologous to human and gut flora. BLAST against the drug bank database yields druggable targets. BLAST can be utilized to identify members of gene families and can be used to infer functional and evolutionary relationships between sequences. The parameters to be considered in analyzing BLAST output are sequence identity, e-value, and bit score. The percentage of matches of the same amino acid residues between two aligned sequences is termed as sequence identity, and for a protein sequence to be significant, the identity should be greater than 35%. E-value tells if the sequence matches are purely by chance, hence, lower the e-value, more significant is the sequence match. 0 or negative value of e-value tells if it is significant or not. Another statistical indicator is the bit score. Higher the bit score, more significant the match is. The bit score measures sequence similarity independent of query sequence length and database size. 100. The BLAST has 4 different types. The BLAST nucleotide search looks for more distant sequences. The BLAST P type can do a protein sequence comparison, and its algorithm is the basis of many others. The BLASTs can search a nucleotide query that can be searched against a protein database, translating the query on the fly. The T-BLAST can search a protein query against a nucleotide database, thereby translating the database on the fly. 27. JTID, bmcnip.in web link. TID is a standalone software for drug target identification. TID depends on the supposition that protein must be vital for the survival of the pathogen and must be non-homologous to its host for it to be called as a putative drug target. TID removes paralogous sequences. It can select the important ones and eliminate the proteins which are homologous to host organisms. It can perform non-homology analysis on gut flora, to identify targets which will not harm the harmless gut flora. Interactome analysis, choke point analysis, pathway analysis, functional annotation, and subcellular localization prediction can be done using TID. It takes less than 2 hours to find putative drug targets from bacterial proteome with approximate 5,000 proteins. 39. Due to these qualities, it becomes a sought-after and a very beneficial tool that is available at bmcnip.in web link. Hence it is a useful tool for rational drug design. It is available at bmcnip.in web link. S. Came Genome Subtractor, 202.120.12.134 web link. The Genome Subtractor is a tool based on the web for analyzing multiple bacterial genomes for a parallel in silico subtractive hybridization. It does a MPI BLAST, which is based in silico subtractive hybridization and generates a list of conserved fragments by matching selected closely related genomes. This can provide clues to the specific environmental adaptation, phenotype, or the bacterial disease. The Genome Subtractor is capable of running rapid blast searches of the multiple subject genomes at DNA or amino acid level against the segmented reference genome, that too within few minutes, mainly because of its parallel computing architecture. The Genome Subtractor provides a very flexible and sliding window-based genome fragmentation approach that can be used to identify short unique sequences within or between the genes. It can also compare protein coding sequences within them. It enables searching or exploring identified core and accessory regions, virulence factors, or bacterial essential genes, including searches against databases of mobile genetic elements, dinucleotide distribution bias, examination of G+C content, and an integrated primer design tool. By providing powerful schematic output for it, Genome Subtractor can, on the basis of available genomes, provides the ready definition of species-specific gene pools. It's a very efficient oligonucleotide design tool, helps in the development of pangenomic arrays very easily. This oligonucleotide design tool is a very simple high-throughput in silico subtractive hybridization analytical tool. The tool supports the ever-increasing number of studies on comparative bacterial genomics that intend to define genomic biomarkers of an evolutionary lineage, their pathotype and phenotype, their environmental adaptation and/or disease association of different types of bacterial species. 40.1 does not need to log into the Genome Subtractor for usage, plus it's also available for free at 202.120.12.134 web link. LTMHMM, www.cbs.to.dk web link. TMHMM is based on hidden Markov model and is used for the prediction of transmembrane helices in protein. It can forecast 97-98% of transmembrane helices accurately. It can also distinguish between the membrane and soluble proteins specifically with a rate of 99%. It also functions with high accuracy that helps researchers predict in the genome the integral membrane protein. On the basis of this, the researcher can estimate that around 20 to 30% of all genes in all genomes encode a membrane protein. It predicts transmembrane helices and discriminates between soluble and membrane proteins with a high degree of accuracy. 41. Users can submit as many as 4,000 protein sequences in FASTA format each time. TMHMM is available at www.cbs.to.dk web link. The organism in whose subtractive proteomics has been applied for drug target identification since 2008. 6. Subtractive proteomics has been used in a large number of organisms. Here we are only mentioning those which have been published after 2008. Subtractive proteomics has been used in two types of organisms, multi-drug resistant organisms and those for whom no effective drug is available. 7 authors' insight on the topic. It is a fast and cost-effective method of target discovery. In this method, proteins non-homologous to host and gut flora is found, which helps in finding those targets that are exclusive to the pathogen. Inhibiting such targets by drugs will help avoid the toxicity issues and will further enable a cost reduction of ADMET evaluation of newly found drugs. 97. It shortens up the time for immune informatics-based epitope prediction, thereby speeding up the time for peptide design. 98. This method just requires proteome data for a start and keeps shortening data at each step. It is a low-cost method and tools mostly used are freely available. Identification of essential proteins in pathogen can be validated via mutagenesis studies. 97. Although this method has several advantages, it also has some disadvantages. This method requires the entire genome and proteome sequence and cannot be used for those organisms whose genome is not sequenced. Targets found using this method requires experimental validation. Essential genes are found using DEGBLAST. This database is continuously updated. With respect to time, the consistency of the number of screened essential genes for a given pathogen is a very noticeable concern. Due to data enrichment of the DEG, this number increases dramatically. 98. Also, we remove proteins 100 amino acid from analysis, but some essential proteins are less than 100 amino acids, so we inevitably miss few novel targets. This cannot be true always. the expression of close gene changes, giving us a false positive result related to the essentiality of protein, within a mutagenesis experiment on the insertion mutation, done in a nucleotide sequence of 300 BP length. A protein that is essential but not homologous to DEG database can be missed. The above method is based on BLAST and therefore one needs to be careful while interpreting results, otherwise a conditional essential protein may be screened and selected during the process. Therefore there is a need to develop a tool that is independent of DEG. One of the findings during the process was that the number of predicted targets gets reduced if we increase the number of different strains within the same genus of the pathogen by using multiple hosts. It is also advised to use all strain-specific hosts in the analysis and use multiple strains of pathogens to identify common targets for broad host range and for all strains as well. 8 Conclusions and Future Prospects. In silico subtractive proteomics is a fast, cost-effective and reliable method for drug target discovery, provided information of genome and proteome of host and pathogen is known. The drug target found by this method needs experimental validation. Multiple analysis is done in this method, requiring BLAST. In order to standardize the method, different BLAST parameters need optimization. It becomes necessary to develop an efficient integrated platform to complete the whole analysis at the same time. And also develop a tool that is independent of DEG, database of essential genes. In order to improve the effectiveness of the original, there is a need for a new approach called in silico mutagenesis and a need to develop a computational validation method. List of abbreviations. CDHIT, cluster database at high identity with tolerance. DEG, database of essential genes. BLAST, basic local alignment search tool. COS, KEG automated annotation server. KEG, Kyoto Encyclopedia of Gene and Genome. Vfdb, Virulence Factor database. Uniprot, Universal Protein Knowledge Base. Consent for publication. Not applicable. Conflict of interest. The author declares no conflict of interest, financial or otherwise. Acknowledgements. The author would like to thank DBT India for fellowship to SM. The authors are grateful to the Research and Development Committee of the Integral University for support.